So this is MISE Lab, Molecular Invertebrate Systematic Ecology Lab of the University of the Ryukus, headed by Professor James D. Ryama. I am Ifena, and this is Yusuke handling the camera. And in this video, we'll be talking about some techniques in the lab, how to use some of our equipment. And we'll be talking about DNA extraction, gel and pipetting technique, and PCR.
And DNA extraction.
PCR and then DNA extraction. And we do this in room B354, which happens to be the molecular room. And please come in with us and learn these techniques as we talk about them. Thank you. These are the DNA extraction kits used primarily in our lab. Both are about the same price, but the Kiagen kit is often used in our lab. There are many other kits and methods of DNA extraction available, so please check the previous study first before you consider the one to use. Thank you. DNA extraction kits contents vary from one brand to another, but they basically perform the same role in the extraction of DNA. And since we did not perform the actual DNA extraction in this video, we'll basically explain to you the role each content of the extraction kit play during DNA extraction. Two major
sorry, go on.
Basically, we use two major DNA kits in the lab. The Kiagen and Monarch DNA kit. So, of course, they do the same thing, and basically the same content. We have the watch, lyse, digest cells, lyse blood, digest blood, lyse tissue, digest tissue, the elute used for preservation of the DNA, the binding keeps all your lysates together. This is the DNA microcentrifuge. We have the low bind, which makes sure your DNA is not bound to the walls of the centrifuge. And like I said, basically the same. They may have different names, but without the same. The buffer, all these buffers definitely perform the same function corresponding to what we have in this other kit, like the ethanol, which helps you wash off things that are not DNA doing your work. And that is it for the kit. You have the tubes. That's all. Thank you. We'll show you how to operate the pipette. And we have to do it attaching the tip, setting the volume, aspiration and ejection. Remember to always wear your hand glove when doing that. First, we'll explain how to set the desired volume or the volume of interest in a pipette. To set the volume, first turn the push button to the desired volume. To set a lower volume, leave the button as it is. However, if you wish to increase the volume, turn the pipette to the desired value after the pipette scale has exceeded the desired setting by half a turn. Next, we'll show you how to install the tips. When attaching tips, we recommend attaching them directly from the rack. Also be careful not to apply excessive pressure to the tip or pipette. After aspirating the liquid, wait a few seconds without removing the tip from the liquid. Take care to aspirate slowly, especially when the liquid is highly viscous. The following are some of the points to keep in mind when installing the tips. Be sure to attach the tip vertically. Thus, attaching the tip at an angle or tapping it may damage the nozzle cylinder. And when you want to remove the tip, be sure to use the ejector button. As explained previously, if the tip is removed from the liquid after aspiration, there will be air bubbles and the proper volume would not have been aspirated. Ensure aspiration is done vertically and not at any angle when taking in the liquid. These are the pipette stands, and these ones here are undergoing sterilization in the UV vent, right? Okay, so basically when you're done using the pipette in the lab, you have to sterilize it. They are both in the molecular room 354.